chemistry would have transitioned to biology on early Earth, which what I mean is how non-life would have transitioned to life on early Earth. So let's using rigorous experiments to start years ago, right? Prior to that, what is it that would have set the stage? One of the important things that you need to try and understand this whole process is to, first of all, try and figure out, would there have been molecules pertinent to biology? Would they have been present on the early Earth? And this is a slide which answers this question. It is thought that there are two ways in which these molecules would have been present on the early Earth. One is by exogenous delivery. Basically, these molecules can ride on meteorites. I'm talking about amino acids. I'm talking about simpler chemical molecules. They can ride on meteorites and asteroids, and they keep bombarding our planet to date. And there's supposed to be a very early time in the history of life on Earth when these things heavily bombarded the Earth. So basically, a lot of this chemical inventory that was important for the transition from chemistry to biology could have been stuff that came from the interstellar medium, where a lot of chemistry is very readily possible. The other important way in which the chemical inventory on Earth became so expansive is also by the kind of terrestrial syntheses that is facilitated in interesting chemical and thermal fluxes can be sort of facilitated in these pools, and these allow for interesting uphill reactions which are extremely crucial for the transition from chemistry to biology to happen. So yes, biomolecules on the Earth would have been possible in these two ways. So given this, one of the big questions then that we come to is to try and understand, how did life come about? So I have a slide here. Unfortunately, I'm not giving this talk in person. Otherwise, I would have paused and asked a question. But you're basically seeing a whole panel of images, including Nelson Mandela, T-Rex, mushrooms, giant sequoias, a catfish, and here is a microbe. What is common to all of them? One thing that is the least common denominator of all of these images is cells. Cells are the fundamental units of all life that we know of. And if you look at these cells, there are two kinds of cells predominantly. You have the prokaryotic cell and you have the eukaryotic cell. And I'm not going to go into details of it, but suffice to say that whether you're looking at a very primitive, quote unquote, primitive kind of cells like the ones you find in bacteria, or you're looking at the more complex kinds of cells which exist in your plants, humans, right, via eukaryotes, fungi, animals, and plants, it is very complex to begin with. I mean, I'm sure you can appreciate how it is probably extremely difficult to envisage concocting such entities from fundamental molecular units. So then, what do we, and one other very important thing about why cell is, I mean, these unicellular entities are super cool is the fact that they might seem like single-celled organisms, particularly the microorganisms, but they can habitate extreme environments present on the Earth. These are called extremophiles. Unlike you and me, they can resist very high UV radiation. They love hot temperatures. When I say hot temperatures, you and I will be bones at those temperatures. I'm talking about 55, 65, 75, some 95. And there are also organisms that can tolerate close to 110 degrees centigrade. Yeah, they can also tolerate very high amounts of salt. Permafrost-dwelling organisms are those extremophiles that can live at very low temperatures. So if you look at where life thrives on Earth today, you can see that every possible niche that you can imagine, despite the extreme parameters that are inherent to the niche, you can see unicellular organisms have habitated that niche. So it's sort of mind-blowing to think that single cellular entities can actually do something like this. So there is this notion of, I'm sorry, give me a second. My mouse is doing something weird. If you look at all the organisms that are present on the Earth that we understand and we have been able to characterize today, they can be put on this tree called the Tree of Life. Here is a cartoon version of it, which is showing that the current known three domains of life, which is bacteria, archaea, and eukaryotes, all of them can trace their sort of ancestry back to something here that has been referred to as the LUCA, which is the last universal common ancestor. And prior to that is where the transition from chemistry to minimal life might have happened. Now, this narrative is a bit old because increasingly it is becoming very apparent to people who work, microbiologists who work in the realm of trying to understand extreme life, microbes in general, and their metabolism, and how they are able to do a lot of very, very cool things despite being really, really, really tiny. It's increasingly now looking like this is how the Tree of Life might look like. A lot of crosstalk between all the three domains. But importantly, going back down, it might have been a common ancestral community of primitive cells, which eventually led to what we see as life today, led to all this beautiful sort of explosion of life that we see today. And this common ancestral community of primitive cells is what is interesting to us in astrobiology, particularly to people who are scientists like me, who are very interested in bringing the individual building blocks together and concocting this ancestral community of cell in the lab. So this is an artist's rendition of how this primitive cell or proto-cell, the first cells, might have looked like. If you look at it, this is like a tennis ball with a slice cut open and you're looking into it. So there is a membrane. And just like your modern cells, and if you look at all of life, it is cellular in nature, excepting for viruses, and I'll not get into it in great detail. But if you look at this membrane that I'm talking about, the cell membrane, it's made of a class of biomolecules called lipids. And the lipids in our cells are very, very complex, but it is thought that very early on in these protocells, you still had lipid molecules, but they were much, much, much simpler. And those particular molecules were readily available on the early Earth by those two processes that I talked to you about early on, exogenous delivery and terrestrial synthesis. And these protocell minimally encapsulates a genetic material that is capable of encoding the information of life and basically replicating, and some minimal metabolic network that will allow the cell to survive, remain far from equilibrium, basically. And this is an even more simplified cartoon version. And this is a very important quote that I want to sort of give you people a sense of. This field started somewhere in 1950s, and I'll get to that in a second. But since then, the best we can ever do is to draw up the story, which is consistent with all the evidence that is being gathered, right? Experiments in chemistry, what we know about the geological processes that might have played out on the early Earth, what biology reveals about the oldest forms of life at the base of the tree of life. Based on all these different evidences, we can kind of piece together this puzzle. And after a century of this fractious effort, the story is finally coming into view. Do we have every single piece of the puzzle figured out? No. But do we understand a lot more about this process than we did in the 50s and prior to that? The answer is yes. And let me walk you through some of that. But before that, I want you to appreciate the interdisciplinarity of this field, right? Like I said, it feeds off of various areas of natural sciences and mathematics. I'm sorry, I didn't put math here, but it's obvious. Math is everywhere, right? Astronomy, physics, it's everywhere. So astrobiology you could be essentially a student with interests in any of these various fields. Any of these various fields, would have pursued it in your bachelor's and would have sort of narrowed down some of the interests to a finer detail and would have done something at master's level, but you still, in your PhD, can be associated with the field of astrobiology by asking relevant questions in the realm of those sciences where you have the expertise, where you have the tools. So what all tools do we use in our lab? The kind of tools we use in the cool lab include tools from biochemistry, from molecular biology, biophysics, organic chemistry, inorganic chemistry, geology. So fundamentally, it is at the edge of geochemistry, geology, chemistry, and biology. We work at that interface. And importantly, we are just not scientists who do experimental work. We also collaborate extensively with those scientists who do research on the computer. Yeah, or who use pen and paper to do research. So there are lots of scientists who do modeling. So we also work in collaboration with them, though we are not experts at that, but we also engage with scientists from some of these various fields to be able to pose the questions that we are very interested in, which is, how did you go from the prebiotic soup of molecules to a protocellular space where you had a whole community of these cells interacting with each other? I'm not going to touch upon this too much, excepting for giving you some sort of glimpses of how we think about when events related to this transition happened. But yeah, the biggest realm is here, where you're trying to understand how do you go from the prebiotic soup of hodgepodge of molecules to a space where you have more biologically relevant systems, which basically allowed for the transition from chemistry to biology on the early Earth. This is the one that I was talking to you about, the early history of life on Earth. So this particular slide sort of goes back all the way to 4.5 billion years ago, which is when Earth is thought to have formed. It evolved a stable hydrosphere by 4.2 billion years ago. And all the interesting things, these are all rough timelines. Yeah, they seem to be changing with every new piece of research article that comes out there. And still there are lots of debates about certain things. But very broadly, this narrative holds, at least for us to come up with valid hypotheses to pose our questions. So about 4 to 4.2 billion years ago, it is thought that very interesting chemistry was happening that is relevant to the biology that we see on Earth today. And between 3.8 to 4.2 billion years ago, a lot of this chemistry allowed for the emergence of a minimal sort of a life space. There's a very important hypothesis in the field called the RNA world hypothesis. And what it posits is the following, that modern life uses proteins, DNA, and lipids and carbohydrates, all of these classes of molecules, right? But very early on, it is thought that the primitive life forms would have mostly used just the RNA molecule. So if you remember the definition of life that I talked about, it needs to be able to encode information. RNA encodes information. Your SARS-CoV-2 virus, which has caused havoc in all our lives and continues to still cause havoc, is an RNA virus. The information of the virus is encoded in an RNA. Not only that, RNA does something very cool, and I told you early on that I'll get back to it later in the talk, and this is where I'm going to get back to it. I'm going to talk about it. RNA does something super cool, which is it can do this engineering kind of functions like your proteins, which basically allows you and me to have this conversation today, right? RNA can catalyze reactions, can do functional, they are also functional molecules, not just informative molecules. So if now you can break down the problem to try and understand how do you go from this prebiotic chemistry space to a minimal life space where RNA sort of encapsulated protocells came about, one would have understood a lot about this primary transition from non-life to life. So where might have all these interesting reactions happen? The reactions where you go from simpler molecules to more complex biomolecules. Depending on whom you talk to in the field, they'll either say that, hey, it happened in terrestrial hydrothermal vents. These are essentially volcanic zones at the bottom of the ocean. Yeah, depending upon another school of thought, they would have happened in these terrestrial geothermal pools, which are basically terrestrial in nature, and they are at the edge of volcanoes. The one important least common denominator between when I say one important, the few important least common denominators of these two niches are the fact that they can facilitate very interesting chemical and thermal fluxes, which is very important for you to go from simple molecules to a string of molecules. So typically, if you look at all of biopolymers, they are, excepting for your lipids, they are polymers in nature. Your nucleic acid is a polymer. It's made up of fundamental entities called nucleotides. Your protein is a polymer. It's made up of fundamental monomeric units called amino acids. Your carbohydrate is a polymer. Yeah, all of these are polymers. So it's extremely important to think about processes and niches that would have allowed for one to go from this monomeric space to the polymeric space, because that seems to be central to this transition. So both these niches are thought to have facilitated some of these reactions, though there are sides and downsides to both these niches. I kind of belong to the group of thought that thinks that terrestrial geothermal pool would have been a much more readily feasible niche in which these reactions would have happened. So I'm going to restrict, for the sake of this talk, I'm going to talk about some of the more finer processes that are facilitated in these conditions. But that doesn't mean these are not interesting niches, the hydrothermal vents. They're very interesting niches. And if you actually sort of take a submarine and go down to these vents, you will see that they're thriving with life even today. Yeah, very complex life even today. So do these niches exist on Earth now? Sorry, I'm using two examples of these terrestrial geothermal pools from this is from Russia, this is from US. They exist even in India. Unfortunately, we do not have too many of these pristine looking sort of niches. We do have a good number of hot springs in the Ladakh region, but a lot of human activity has already contaminated a good bit of these hot springs. But having said that, these hot spring-like, geyser-like terrestrial geothermal pools are very readily even present on Earth, even to date. And if you go and actually do some field work there, which we also do in the lab, you can actually, this is called analog research, where you're trying to go to a field site which is reminiscent of some of the conditions that you're trying to replicate in the lab and try and do experiments there. But the one important thing, the reason we do this is these environments are super complex. What we're trying to do in the lab is to use a very reductionist approach. We're trying to sort of minimally parameterize the space and work in that space. because otherwise it becomes too complex, too quick, and you don't understand what's happening. But it is also important that once you've understood how the minimal processes work, how they sort of pan out, it is also important to challenge yourself to go out into the field site and bring back samples from there and work in the lab, or also conduct some of the experiments which the field experimental stations might allow right at that venue, to try and understand if the processes you have been sort of using a very reductionist approach to understand in the lab actually hold good even when you are working with analog conditions. So there's a model out there that talks about how these protocells might have emerged in these terrestrial geothermal pools. So if you had the monomers that are pertinent to the formation of the protocell, what are the two monomers that you need? A few monomers that you need for the formation of protocell. You need the monomer that can form the membrane. You need the monomer that can form the informational molecule. We are going to now sort of say RNA is that informational molecule to make life easy for ourselves. And you will also need a bunch of monomers that can come together, form networks that will then provide energy to sustain these protocells in those local environments. So here is the edge of a tidal pool here, yeah, or a geothermal pool. If you have the monomers pertinent both to the informational molecule and to the boundary molecule, you can sort of have these, the very cool thing about, you see these little squiggles here, those of you who have done a little bit of chemistry will appreciate that these are hydrocarbons, yeah? Basically, you have carbons and hydrogens in this chain, and the head has polar molecules. So these molecules are called amphiphiles because they have a water-loving head and a water-hating tail. And as soon as these molecules, like these shown here in this white ball and stick here, right? As soon as they hit water, all the water-hating groups will bunch together, like it's shown here in the inset here, this cartoon here. These black sticks are the water-hating groups. These colored heads, the green and the turquoise, are water-loving. So as soon as you put a bunch of these molecules in water, all the water-hating groups will come together so that they can exclude water, and the water-loving groups are on the outside. What this allows these molecules to do is to form these higher-order structures. Here are three of them. They can form micelles, they can form bilayers, and bilayers can fall back on themselves and form very interesting containers called vesicles. You look at the micelles here, sorry, it's not very clear, but the water-loving head group, the white ball is facing the water, and the water-hating group is on the inside. In the bilayer, you see that the tails are towards the inside and the water-loving heads are towards the outside. And when you form these long enough stretches of bilayer, they become unstable, fold back on themselves, and very quickly form these vesicles, which, if in the vicinity had other interesting monomeric molecules, it could entrap it within itself. It could just engulf it within itself. And then, just like on modern Earth, you would have had wet-dry cycles, which would have been very readily facilitated on the early Earth. Day-night cycles, yeah? Early Earth is thought to have been much more hotter. High temperatures, a lot of heat, so dehydration. At night, temperatures are much lesser, things cool down, you have condensation, and you have rainfall-like scenarios, wetting cycles, right? So these kinds of wet-dry cycles can be facilitated by day-night or diurnal cycles or seasonal changes, or at the edge of tidal pools by just tidal activity. When tide comes in, it's a wet cycle. When the tide goes out, it's a dry cycle. These kinds of wet-dry cycles have taken a very prominent center stage in the field, predominantly because they can facilitate very interesting polymerization reactions, which are otherwise not possible in aqueous medium. What do I mean by this? You want to bring two of these monomers, like your nucleotide monomers that make up the DNA, or the amino acid monomers that make up the peptide. You want to bring the two of them together and make them interact. They hate doing it because they are thermodynamically very stable in isolation. So you need to force them to interact with each other. And fundamentally, the polymerization reaction is a condensation reaction, which means you're pulling out a water molecule. So with an abundance of water in the surroundings, this reaction is not going to happen. But the second you reduce the water activity by invoking these naturally occurring dehydration cycles. So these higher order structures, they can collapse and form these multilamellar sandwiches. This is a cartoon rendition, but this is a real freeze fracture electron microscopy image of these lipid molecules in dehydrated state. They can form these multilamellar sandwiches. And if you have these red balls, which depict the monomer, so let's call them the monomers of RNA, if they're present in the aqueous solution, they were engulfed in that tennis ball to begin with. And on dehydration and forming these multilamellar sandwiches, they can now come into close proximity with each other. And within these layers, you have very little water activity. So you can now force these condensation reactions to happen. Basically, you're driving the reaction towards polymerization, even though their favorite state is to be in single state. They're very happy being single. They don't like to mingle. So you have to force them to do that. So these wet-dry cycles are thought to have facilitated formation of these informational polymers, and multiple such dehydration-rehydration cycles. Oh, I need to talk about a subsequent rehydration cycle and what implication it has. So now you already see formed these longer polymers, in these spaces. On a subsequent rehydration cycle, either due to rain or due to cooling down during the night times, you can actually have these multilamellar sandwiches once again bleb out to form these protocell-like entities. And this inset, again, is a fluorescent microscopy image of synthetic vesicles that can be readily formed in the lab. And the very dark fluorescent green color that you see in these tubes and in these things that are blebbing out of the tubes, the bulbous structures, that's actually an encapsulated fluorescent molecule. So you can have these kinds of processes very readily happen during a rehydration stage, which is thought to have led to the formation of these protocellular entities. And multiple such cycles is thought to have allowed for the formation of the most sturdy protocells that would have essentially survived under those harsh conditions. So currently what I'm talking to you all about at some point was completely a hypothetical sort of a problem, right? Or it was based on a hypothesis. And this was propounded by two people who lived in two different parts of the world. And back then, no internet, no nothing. They were snail mails, and they probably could communicate between Russia and America over a period of months, right? So this is Oparin, and this is Haldane. Oparin basically was a Russian scientist, Haldane an American scientist who has a very interesting India connection. Both of them posited that on the early Earth, your oceans, your primitive oceans provided the laboratory setting for all of these interesting reactions to happen. Where did the energy come from? The energy could have come from lightning in Earth's early atmospheres, or it could have come from ultraviolet radiation. There's a reasonable knowledge about how ultraviolet radiation would have been much more pronounced on the early Earth. So given these high energy conditions and the presence of a lot of these interesting molecules like methylene, ammonia, dicarbon, carbon dioxide, hydrogen in water, in your primitive oceans, you could have they can basically interact with each other using the energy from these sources to form spontaneously molecules which are organic now. Here is a chain of amino acids strung into a peptide. So this was a hypothesis that was sort of posited by both Oparin and Haldane. And Haldane is attributed with coining the term primordial soup, where he basically said that all of this interesting phenomena were basically allowed for the formation of relevant molecules that would have been sort of present in your primitive Earth in various sort of aqueous bodies. A theory or a hypothesis is a hypothesis till it is proved otherwise. And when did the first evidence for this not just being a hypothesis but plausible scenario, when did that come about? That came about in 1953. This is the very famous Urey Miller experiment, where these people recreated that primitive ocean here in this particular contraption. This is your primitive ocean. You see here it's mentioned as water. And you had all these ingredients, right? Methane, ammonia, dihydrogen. And when you heat them up, early Earth was very hot. When you heat them up, they vaporize, and the vapors go into the space where there was this electrode contraption set up, and they could provide a spark discharge of very high energy, which then allowed very interesting reactions to happen in the space. And once the vapor comes here, this is a condenser where you once again will have this gaseous phase turn into liquid phase, and this particular cycle can keep going on. And whenever you want to retrieve samples, you can retrieve, oh goodness, what happened? Sorry about that. Whenever you want to retrieve samples, you can retrieve samples from this particular space and analyze them. So with limited technology that they had back then, access to limited analytical techniques, the two of them, particularly Stanley Miller, who's right here, he was a postdoc of Harold Urey, he went on to show that from this bunch of inorganic molecules, you can form organic molecules relevant to life, which is glycine, glutamic acid, alanine, serine. Sorry, here the E's are all down, but please understand that that's what it is. Urea, for the first time, urea was for the first time demonstrated by a very famous scientist much before this in the late 1800s, Frederick Wöhler. But having said that, all of these molecules could very readily form under these conditions, showing that you indeed could have the operand-Haldane theory actually demonstrated in an experimental setup. This set the stage for the field of science called prebiotic chemistry, which is something that we do extensively in the lab. And suffice to say, since 1953 is very important for another year. 1953 was when your DNA structure was sort of demonstrated by a few important scientists. Only a subset of them got the Nobel Prize and left Rosalind Franklin out. I won't get into that story, but 1953 year was a landmark year for many reasons. Since then, up to now, where are we? We have sufficient understanding of how we can go from this primordial soup of fundamental ingredients to precursors that can form amino acids and proteins, to precursors that can form nucleosides and nucleotides, and to precursors that are important for the membranes. Yeah. So we have been able to get to this point in all of these years. However, we still haven't been able to systematically demonstrate the formation of a living protocell in the lab. So, like I also told you, one of the other important goals of astrobiology is to try and understand the origin and evolution of extreme life on Earth too, because it helps us to use a top-down approach to try and understand how you can sort of go from how you can use the information that you glean from some of these extreme microorganisms to try and understand what the base of the tree might have been populated with, what kinds of entities they might be populated with. So again, your protocell, which are essentially fundamental lipid membranes encapsulating some form of a genetic material, here an RNA and some minimal metabolic network. And then there is this space where it's thought that it was populated by the common ancestral community of cells. We do not have proof for it because the kind of, it's very difficult for these things to leave any markers. There are certain sort of geological markers that we can look at to try and sort of glean something in a sort of indirect manner, but you wouldn't, for example, have a fossil of a protocell. It's extremely difficult for that to those to be preserved under those conditions. But here is where we get to this very important point in the early history of life, where the first complex organisms came about, the first complex unicellular organisms came about. Yeah. So this is an example of a species of microbes which comes under cyanobacteria. These single cell entities are essentially thought to have been the most important players for making Earth habitable for more complex life forms. All the stuff that I talked about to date, when I say to date, till now, were processes that were facilitated when Earth had absolutely very little to no oxygen. It is these organisms that came about, and they basically facilitate a process called photosynthesis, which essentially is thought to have oxygenated the environment, the Earth's environment, which allowed for more complex beings to come about. How do we know about these organisms? This is origin of prokaryotes and the oxygenation event that I talked about. Some of the oldest evidence of it goes back to about 3.5 to 3.9 billion years ago. The latest evidence is from a hot spring deposit, which is close to 3.87 billion years ago. And it is also from the Western Australian part of Perth. You have these beautiful deposits. These are called stromatolites. These pillow-like structures form as a result of interaction between microbes and earth and clay and dust. Basically, it results from an interplay between microbiology and geology. Yeah. So basically, what these things are is, if you sort of slice them, you will see these alternate black and golden striations. Yeah. The black striations are indicative of the geological processes, and the golden striations are indicative of the biological process. So these are some of the oldest evidence that we have of life, and it goes back all the way to 3.9 9 billion years ago. This is still a little outdated. And it's the single cell organisms present in these golden striations that are thought to have oxygenated the Earth's atmosphere. They use the sun's energy, and they use water, and they use carbon dioxide, which was very abundant on the early Earth, to basically fuel their existence. And as a byproduct, they released oxygen into the atmosphere. And with millions of years of this process, you eventually had oxygenation happen on Earth. Now, when did eukaryotes, or very old cousins of ours, emerge? It's thought this event must have happened about 2 billion years ago. So here is this clock-like image, right? Midnight is here, you have morning here, noon here, afternoon here. So we were talking about some of the oldest known rocks, geological evidence we have of it. We can use sort of zircons as an indicator to try and understand this. Some of the oldest known rocks go back all the way to 4.2 billion years ago. Where do we have the oldest fossils that are present? I think this can go back now. This thing has to be slightly updated. This can go back all the way to 3.8 billion years ago. So oldest definite fossils of prokaryotes for sure, 3.9 billion years ago. But it's about here in the 2 billion years ago, you actually have the first fossils of eukaryotes. Eukaryotes are more complex cellular structures which are thought to have emerged as a crosstalk between bacteria and archaea. I showed you the 3 domains of life. You have bacteria, you have archaea, and it's thought that the symbiosis between bacteria and archaea is thought to have led to the emergence of eukaryotes. And there is some very interesting microbiological evidence for this hypothesis. This was proposed by this famous scientist called Lynn Margulis. Lynn Margulis was the ex-wife of Carl Sagan, who was known for, who was a very famous cosmologist and astronomer. So these are thought to have evolved into more complex multicellular entities, which started talking to each other and doing things in conjunction and forming more complex structures. Without getting into too many details, what I'd like to tell you is I've given you some sense of the steps leading to the emergence of the primitive cellular entity or the protocell. And in our lab, we are interested in every single step that is involved in this process. Basically, we're interested in the fundamental prebiotic chemistry to try and understand the niches and the parameters in the niches like pH, temperature, presence of certain kinds of solvents. What implications do they have for the formation of building blocks? And next step, we're very interested in understanding how this primitive building blocks or fundamental building blocks, sorry, I'll take away the term, it's wrong to use the term primitive. They're complex enough, the building blocks. How do these building blocks come together to form much longer polymers that then allows us to now get into the space or the space where minimal life starts to emerge? Then if in this RNA world, which I talked to you about, where primitive organisms that used protocells, that used RNA as the information coding molecule, if you did have this RNA world happen, and there is reasonable proof in biology today to show that this must have been a very important stopgap during the origins of life and evolution of life. And one of the important evidences for that is the fact that the core of your ribosome, which makes the proteins or the workhorse of your cells, is actually the core of the protein. Protein, when I say it is a polymer made up of amino acids, but the core of the protein, which does the actual stitching up, core of the ribosome, which does the actual stitching up of the amino acids to form proteins, is made up of RNA. The fundamental peptide bond forming reaction is facilitated by RNA, and it is extremely important that the protein present around it is also there for it to basically facilitate this fundamental formation of this bond, which allows us to do what we do today. So it is also one of the important things to think about is very early on, how did we have RNA-based catalytic entities that might have done some of these reactions, but in a much more haphazard manner, and it might not have been fantastic, but there are advantages for this process to not be perfect either. But that is another thing which we're very interested in understanding. And finally, we're all super invested in understanding this membrane business here, right? This particular container's outer boundary is fascinating to us. How did the molecules that form these membranes come about? What is the kind of complexity that might have been present on the earlier? Why do we see what we see today? What might have been, are these things good enough if we now move to more extreme conditions that might have been prevalent earlier. So then that makes us want to understand what are the possibilities that are out there, chemically speaking, that might have allowed us to form very robust protocellular compartments. So these are the kinds of things which we are very interested in understanding in the lab. We do a lot of things, not in isolation. Science doesn't happen in like I already pointed out, astrobiology is a field of science which is heavily interdisciplinary in nature. So we have to work with people from diverse backgrounds, theoretical chemists, experimental chemists, geologists, planetary scientists, cosmologists, people who do modeling. There's a lot of very, very, very interesting sort of tools that we can use to address questions in a much more robust manner. So we are sort of looking at a lot of these questions that interest us in conjunction with people, scientists who are experts in their domain, not just from India, but across the world. And I'll finish off with this particular thing. Why is astrobiology suddenly very interesting? Other than the fact that astrobiology is just fascinating, right? How did we come about? Of course, we are super complex. How did the much more simpler organisms come about? I only showed you that evolution of life on Earth up to 2 billion years ago. After 2 billion years ago, it was an explosion of complexity in the life forms that we see. You had this Cambrian, pre-Cambrian era where you had a lot of interesting environmental constraints which allowed for an explosion of complexity in life forms. So it's called the Cambrian explosion, which is thought to have been a period when the maximum number of life forms would have existed on the early Earth. And after a lot of evolution, you get to the space where about 65 million to 250 million years ago, you had the age of the dinosaurs. I was talking to you about unicellular organisms, which existed 2 billion years ago. But between that time and around 250 million years ago, you already had super complex living beings like dinosaurs walking the Earth. And if you looked at that clock, very close to midnight is when we emerged, literally 200,000 years ago. Prior to our emergence on this planet, there was all of this grand drama that was happening, of which we have very little understanding. And we have subsequent understanding about what was happening several billion years ago due to multiple logistical constraints, but also due to the fact that it's sometimes very difficult to constrain some of the parameter spaces that might have been plausible back then to be able to systematically understand some of the processes that we wish to understand. Nonetheless, the field has come really far, and there's been up to 49 missions to date. None of these have ISRO in the picture. These are all either NASA or ESA or the Japanese space agency called JAXA and other. These are sort of the three most prominent space agencies, and they've had multiple missions to different planetary bodies, and a good number of them had some minimal experimental modules that they carried, which was important for understanding astrobiological processes, but nothing like Mars 2020 Perseverance rover. Your Perseverance rover, which made a splash just a couple of weeks ago now, maybe, I don't know, close to a month, maybe, or still close to 3 weeks, maybe, it landed on Mars, right? It's a lot like its cousin Curiosity rover. It looks a lot like Curiosity, but it's just been embellished with a whole bunch of gadgets and equipments that's going to allow it to do mind-blowing science, fundamentally to understand if there was ancient microbial life on Mars. For example, your MAST Cam Z, it's right here, this zoomable panoramic camera, right? It's a system which allows you to take panoramic images. And other than that, it is also sort of rigged with contraptions which can allow you to determine the mineralogy of Martian surface. But mainly the MASCAM is going to be able to help with giving the rover instructions as to where to go, where to move. Perseverance landed in a crater called the Jezero crater. Jezero crater is extremely interesting as one of these analog of these terrestrial hot springs that I was talking to you about. It's not the only crater on Mars. There are so many other craters, but Jezero crater is super cool in the sense that it has this gorgeous delta that has still been preserved. You can see it even in images. And there is a lot of hope for being able to sort of try and see if ancient microbial life, if it did exist there, there is a good chance that it could have left some signs of it for us to find. So the question is, will we find them? You have the SuperCam. SuperCam is like right here, right? It's a laser-based microimager. It's going to be able to allow you to detect the presence of organic rocks in organic compounds in rocks, and from a distance. Once you have some sense that, oh, that particular rock has some interesting organic matter, you have now much more fine-tuned instruments which will do the rest of the stuff. You have this very interesting contraption called the PIXL, Planetary Instrument for X-ray Lithochemistry, right? It's essentially an X-ray-based fluorescent spectrophotometer, contains an imager. It allows you to get the detailed elemental composition of these Martian materials, your rock, your Martian dust, and it can kind of look at it and it can actually figure out the elemental composition of these entities. And then you have something called Sherlock, something which I find this acronym very cute. Scanning habitable environments using both Raman and luminescence to look at organics and chemicals. And this is essentially a contraption, a spectrophotometer that is going to use a UV laser, ultraviolet laser. Again, will allow you to do some detailed mineralogy, try to understand the elemental composition of the materials that it's going to deal with. And it also has this drill bit, which will allow it to basically drill into very interesting there's a whole bunch of people who are systematically trying and understanding the data that's coming from all of these different instruments. And finally, if they've zeroed in, say, onto that beautiful part of the delta, which has some very interesting and very promising elemental profile, they can actually use this drill bit to dig into it, to drill into that space, get samples out, seal it into tubes, which in future, they're expecting that in the next 10 years, there's going to be two other missions, which will allow for us to retrieve these samples from Mars and bring it back. So I think that's like mind-blowing. This is where astrobiology is now. And like I said, we do analog research in Ladakh too. And here are images from a 2016 expedition that we did with a bunch of collaborators from US, from Australia. We had a lot of fun. That was the first time we ever did field work, and we learned a lot. We hope to be going back very soon. And here is a slightly oldish picture of the team. But yeah, thank you very much for your time. The origins problem is knotty, but it's not insoluble at all. And I hope next time I come and talk to you, I'll have more interesting updates. And I'll stop with this slide. Thank you for your time.
Thank you so much for this amazing talk, ma'am. We learned a lot of new things. And now, if we can take some questions, if that's okay with you.
Yeah, absolutely.
So there were some questions in chat box. If Varad is in the meeting, can you please raise your hand so that you can directly ask your question to him? Okay, so I will just read.
You might have to unmute Varad.
He was asking, how can undergraduate student join astrobiology, or path should choose after graduation and volunteer your project?
Okay, awesome. So I'm going to use my own story to tell you how I landed in astrobiology. Yeah, I was not born an astrobiologist. Trust me when I say that. So I am a biochemist by training. I did my PhD when I was introduced. I'm sorry, I'm forgetting your name. While I was introduced, he mentioned that I worked on a problem in PhD pertaining to sickle cell anemia. Sickle cell anemia is one of the first genetic disorders that was described. And I worked in National Institute of Immunology in New Delhi with Dr. Roy to try and understand how mutations to different regions of this molecule affect the sickling behavior of hemoglobin. This is a very important problem, mainly because people who are born with this genetic mutation there are a whole bunch of mutations that have been described to date, but there's one very dangerous one, which is that you have 2 alpha chains and 2 beta chains in hemoglobin. And in the beta chain, you have a GLU to VAL mutation, a glutamic acid to valine mutation, which basically completely changes the property of this molecule, and it cannot bind oxygen as efficiently anymore. So people that are born with this disease don't survive beyond their late teens. It is so bad. And only recently, very recently, and it's still an experimental technology, this one lady in US has been given a CRISPR-based treatment to be able to sort of provide a workaround to this problem of hers, genetic problem of hers. But otherwise, the only way these people have any hope in life is to constantly have blood transfusions, because these sickled RBC are the molecules in which you have the hemoglobin. Sickled RBCs have a much shorter lifetime. Their lifetime is not like 120 days, which is the lifetime of a healthy RBC. So I was very interested in looking at all of these problems. And then I got very fascinated fundamentally by protein aggregation as an issue. Good hemoglobin becomes wonky because of aggregation, a process which causes its clumping. And this particular protein aggregation is a problem even in other famous diseases like Parkinson's, Alzheimer's. So working in this realm got me very interested in these other cooler protos. Dr. Fink, he lost a very important big funding. So we were all kind of laid off and reappointed on a monthly basis. And then soon I was told, Sudha, in another month, you need to find another option. I was almost packing my bags to come back to India when a complete set of coincidental events led me to this field of astrobiology. I figured, all of us are fascinated by this thing, right? How did we come about? Is there life elsewhere in the universe? So in that sense, I was super fascinated with this question. What I didn't realize is people can very systematically form very rigorous hypotheses and ask these questions in a much more rigorous manner, like we do in my lab today. So I got intrigued. I said, no harm, let me figure out what this field is. Joined Dave's lab, Dr. David Deemer's lab, fell absolutely in love with the field and continued in his lab for a couple of years, and then went on to Harvard to do my postdoc. So if you look at this, when did I become an astrobiologist? After my PhD, right? So you do not have to be, there's no BSc in astrobiology. There could potentially be an MSc in astrobiology. It is still so interdisciplinary in nature. Everybody can, it's no fun being jack of all trades and master of none. So if you are a person who loves physics, do your undergrad and masters in physics and become really good at that. And maybe you can have ancillary chemistry or whatever it is that you like. Build your tool set, and become an expert in a bunch of tools, and then apply those tools to ask pertinent questions in astrobiology, which happen in various labs in the world. It's still not a very big field, but it is growing. So there are biochemists like me, there are geologists, there are planetary scientists, there are cosmologists, all of them working in this area. And depending upon your expertise and your interest, you can find a relevant lab and sort of try and join that lab. I hope I've answered your question. Somebody, Advait Arvind Joshi, has his hand up.
So before that, there was another question in the chat box.
Sure.
From Anish. This question was related to apparent Haldane theory. He was asking, is origin of life explained by appearance of polypeptides before generic material?
I don't know what you mean by generic material. It's a very vague term to me. Overall, the transition from chemistry to biology can be envisaged as a polymerization problem. One can think about sort of systematically thinking about this transition, but by trying to understand how do you go from a monomer space to a polymer space. So you can be, talking about formation of peptides, you can be talking about formation of DNA, you can be talking about formation of RNA, you can be talking about formation of carbohydrate polymers. Any of these polymerization events can shed a lot of light and can importantly basically give us a lot of understanding about what kind of environmental sort of spaces, niches, what kind of geological sort of parameters, what kind of chemical parameters might have been important for these transitions to happen. The only reason we have a bigger focus on trying to understand the RNA world is because of the RNA world hypothesis. We are not sort of married only to that, because it's very important to realize that your prebiotic soup was a complex mix of a lot of different chemical entities. So to say that when you're trying to understand in a lab setting, how did RNA polymers come from RNA monomers, you're only looking at RNA monomers. The second you complexify the reaction, you're going to now start finding all of these side reactions happening. And for, like I already pointed out, we use a reductionist approach to try and understand the fundamental processes underlying some of these polymerization events. Nonetheless, it is extremely important, even in the lab setting, to complexify your starting soup, to incrementally complexify that starting soup and try and understand how these events that you think might have been very important for the transition from non-life to life, how they might have actually panned out in a more complex scenario. And that is one thing which we do in the lab. It's like shooting ourselves in the foot, but it's a very important piece of thing to understand for better understanding of how these events might have panned out on the early Earth. So yeah, I mean, this kind of constraint within which you want to understand this transition allows you to a reasonable extent to try and understand what might have been the niches and the parameters, chemical and physical parameters that allowed for the transition from chemistry to biology. Oh, is he saying that peptides should have been the genetic material? Is that his question?
Maybe I can ask him to ask his question.
Yeah, I mean, please.
Yeah, so my question was that the Oper and Haldane hypothesis explains that proteins would form on the early Earth conditions, but there must be something about replication, because that's essential to life. And RNA were some types of RNA are capable to replicate, but I'm not sure if proteins are able to replicate by themselves.
Now I get your thing. So yeah, here's the thing. So proteins by themselves cannot do the kind of replication that involves hydrogen bonding based replication that DNA and RNA can do. Yeah. How does your double helix in a DNA form? It's because of this base interaction, your adenine, guanine, thymine, cytosine, and uracil present in DNA and RNA bases, DNA and RNA molecules. These are called nitrogenous bases. They have these exocyclic atoms which allow them to hydrogen bond with each other. That is one very important way of information transfer that happens in nucleic acids. In proteins, unfortunately, you do not have that kind of a capability. However, there is a sort of a small hypothesis that is still doesn't have too much of prebiotically relevant proof, but it does happen in some of these protein aggregation disorders that I'm talking to you about. There is a notion of structural information, not molecular information, but structural information, which is thought to be propagated in protein-based systems. However, I still have difficulty envisaging how molecular information can be coded in a protein-based polymer. It's still unclear to me and to many others in the field. I hope that has somewhat answered your question, Anish.
Hello, ma'am. Thank you for this wonderful session. I had a general doubt that I'm also in contact with Mr. Siddharth Pandey from MIT and Mr. Pushkar Vaidya.
Yeah.
And as you also told that you also did an expedition in Ladakh. Even they are planning. So what makes Ladakh so special for attracting people to do expedition over there itself?
So
not somewhere else.
Ladakh is a Mars analog site. There are lots of these geological features in Ladakh which are very reminiscent of Mars. You have these hot springs, yeah, like the one which Jezero crater is thought to have probably supported. There's supposed to have been water, and temperatures were pretty extreme. So the water should have some sort of a source, right? So this kind of geological feature, which is reminiscent of Mars, is very present in Ladakh. You have these 100 dunes, and they're formed by these physical processes that make sand and dust form these special patterns. These are also found on Mars. There are sort of contemporary geological features that are present on Mars. So these high altitude deserts like Atacama Desert, Ladakh, these are very, very dry, arid areas which have a lot of geological features that are reminiscent of other planets, particularly Mars. So if you want to, for example, send Perseverance rover to Mars, which has happened, some of the preliminary dry runs, you can concoct the rover, but you need to make sure that the rover can traverse the geological morphological features that are found in that space effectively. How do you do it? You can, in principle, take the rover's model to Atacama, to say Ladakh, and you can actually make sure that it is able to traverse the terrain. There are a lot of rocks of various sizes that are present. There are a lot of these features which are extremely rugged. Can the rover traverse these features, for example? These are the kinds of preliminary test experiments that a space like Ladakh allows you to do. In our own, pertinent to our own research, we are very interested in understanding how the RNA world came about. In the lab, we do it under extremely pure buffered conditions. Yeah. But it is very important to try and understand it in much more realistic scenario, which a hot spring site in Ladakh allows us to understand. Like I said already, there's a lot of human activity in Ladakh, which has tampered with the pristine aspect of a lot of these hot springs. But if you still go to the source of the hot spring, which is very, very sort of retains a lot of the geochemical nature of the hot spring, you still can bring back samples with you to the lab and try and do some experiments. So what it has made us understand is the following, that analog environments provide a complexity which is lacking in laboratory experiments that we do, because your hot springs in Ladakh are lined by are lined by different kinds of rocks and different kinds of minerals that leach out very important ions into the water. They can have a bearing on the process that you're looking at in the lab. In your lab, you're not even factoring any of these things, but by going back either to Ladakh and doing some of these experiments in the field, or importantly, bringing back samples from there and trying to work with them in the lab, you can try and understand these processes in a much more realistic manner. So I hope that has answered your question.
Thank you, ma'am.
Sorry, and also quickly for the microbiologists, Ladakh is extremely interesting because it hosts a lot of these extreme sites where you can have extremophiles thriving. Permafrost still exists in Ladakh. And if you can dig deep enough, you can actually bring back samples to the lab to try and understand what kind of microflora exists there. So yeah, there's a lot to learn from those kinds of analog environments. Sanket, the one who introduced me. Sorry, now I remember your name. Yeah, tell me, Sanket.
Yeah, thank you, ma'am, for your wonderful talk. And my question was regarding the silicon-based life. I mean, what are your views on the silicon-based life, and where the semiconductor atoms are the basic building blocks? And can such life evolve by its own?
You know, a lot of, even though silicon seems very close to carbon, the actual physical properties and the chemical capability of silicon-based materials is nothing in comparison to carbon-based chemistry. Carbon-based chemistry is extremely versatile, extraordinarily versatile. Silicon-based chemistry still is not that versatile. So even though it might seem like they are so similar as atoms, in terms of what they're capable of doing with other atoms, the kind of reactions that silicon can undergo with other atoms is way more restricted than carbon is. So for that reason, it is so much, and given the fact that life on Earth, all life on Earth to date, and we still haven't scratched the microbial life, we've only scratched the tip of the iceberg as far as microbial life goes. But whatever we've been able to understand, all the way from microbes to the more complex organisms, there's a reason, it seems like there's a reason why carbon-based life forms were particularly preferred under the geological and planetary conditions that were prevalent on early Earth. So silicon didn't seem to stand a chance, surely under our conditions. In any other condition or on any other planet or exoplanet, because of that local condition there, could a silicon-based life be plausible? But under our early Earth conditions, no. The kind of amazing things that carbon-based chemistry can do, silicon-based chemistry still can't do.
Yeah, thank you, ma'am.
Thanks.
Okay, thank you so much, ma'am. Now, Dr. Raka Dabare would like to say a few words. So, ma'am, I hand it over to you.
I'm sorry, I'll stop sharing. Maybe. Yeah, sorry about that.
I cannot, thank you, Adishri, and thank you, Dr. Sudha. I just had one more question from my side. I wanted to know about the Lonar crater, which happens to be around 35 to 50,000 years old, which is a meteoritic crater. So how far are the Indian astrobiologists working in the crater area?
So a very dear friend and head of Center for Astrobiology in Amity, Aqua, Siddharth, and a few of us are very interested in the Lonar crater. The problem with Lonar crater is you can't do too much microbiology business. It's been extremely heavily polluted by human activity. Super extremely polluted by human activity. However, it still holds promise for trying to understand certain geological processes. And hence, there are a bunch of our colleagues from NASA and from Mars Society Australia who actually are very interested along with us to understand some of the geological processes that might be facilitated in this kind of a meteorite impact crater. So we're just beginning to formalize some of these things, and hopefully there might be an expedition to it in the near future. It's not, I mean, with COVID going on and stuff, nothing is for sure till things settle down. But if everything goes well, we should have an expedition to explore some of these things in a little more systematic manner to say what is the kind of questions that we can ask if Lonar crater was our field site. So it's happening.
Okay.
Thank you for bringing this up.
Yeah, thank you. And one more thing, in continuation with Sanket's question regarding silicon-based life, as far as I understand, if you talk about the nucleosynthesis process which takes place inside the sun, apart from hydrogen, helium, carbon, oxygen are the elements which are most abundant in the universe. So silicon, I don't think, I mean, it's that abundant. It's much less abundant than carbon. So there's no question of having a silicon-based life because that abundance problem comes there, I think. I don't know whether I'm right.
Thank you. No, that is one of the answers that some of the planetary scientists put forth. Since I'm a biochemist who works in the chemistry realm, my answer is more chemistry oriented, but I've heard this, Raka, and I think what you're saying is not wrong. There's another problem too.
Okay, yeah. Thank you so much. And Sudha, I'm really grateful to you because I know in spite of your so many problems you are facing, you finally managed to give us time. And today I would like to tell you that most of our biological faculty from biological sciences have also joined the YouTube channel from microbiology, biotechnology, botany and zoo. And I'm sure you have triggered some reactions in them. So I'm hoping that from Ferguson College we'll have future astrobiologists to come. Thank you so much. And we would like to have you some.
And thank you very much, Raka, again for hosting me. Thanks a lot. I appreciate it. Thank you.
Thank you.
Thanks a lot. I hope it didn't fly over people's head because of all the chemistry and stuff. I didn't show a lot of hardcore chemistry slides or biology slides. It was just kind of to give you an overview of the narrative. So I hope that helped. But I'm happy to take any questions if anybody has. They can mail me or you can compile them and send it to me and I'll be happy to answer them. Best of my ability. Okay. Ciao. Bye.
Thank you, ma'am.
Thank you. Bye.
So we will be having the poster exhibition starting from 4:30. Everyone has been sent the link to the website in the mail. You can see all the posters on the website. We have also sent a link to the Slack workspace, and I would request everyone to join that as well. Any questions regarding the posters will be addressed from 4:30 onwards. So hope to see you all then. Thank you.